Welcome to the Future Lab. I'm your host, Matthew. Today, we're diving into a fascinating review paper exploring how large language models are transforming the field of bioinformatics. It's a field exploding with new discoveries, so buckle up. Large language models, LLMs, like BERT and GPT, initially designed for understanding human language, are now proving incredibly adept at analyzing biological data. Think of it, applying the power of AI to understand the language of life itself. We'll be exploring how these AI tools are accelerating research across multiple areas: genomics, transcriptomics, proteomics, drug discovery, and even single-cell analysis. This is truly cutting edge, and it's changing how we do research. The sheer speed of innovation here is remarkable. Just a few years ago, many of these applications were science fiction. Now, we have powerful tools actively speeding up discoveries and generating new insights. It's a game changer. This episode isn't just about the what, we'll also delve into the how. We'll look at the underlying techniques these researchers use to adapt these powerful language models to unlock the secrets hidden within DNA, RNA, and protein sequences. It's ingenious. So, what does this mean in practical terms? Break time equals 0.5s, greater than well, we're talking about faster drug development, improved disease diagnostics, and a deeper understanding of everything from the smallest cellular processes to the complexity of the human body. The implications are enormous. Before we get into the nitty-gritty of the methods, let's start with a high-level overview of the research itself, and what the authors set out to achieve. We'll then move on to a deeper exploration of the techniques. Break time equals 0.5s, greater than. Are you ready to explore the future of bioinformatics? So, let's talk about the core of today's episode, a review paper by Zhou et al., titled Large Language Models in Bioinformatics, Applications and Perspectives. It tackles a really important area. At its heart, this paper explores how these powerful language models, originally designed for things like translating languages or writing stories, can be adapted to work with biological data. It's a fascinating shift. The reason this research is so significant is the potential to radically accelerate and improve biological research. We're talking about speeding up processes that currently take years, if not decades. The authors focus on the application of LLMs like BERT and GPT across different levels of biological information, what we call omics levels, genomics, transcriptomics, proteomics, and so on. They've looked at how these models handle various types of biological data, and the results? Break time equals 0.5s, greater than well, let's just say they're impressive. They found that these LLMs are incredibly effective in a variety of biological prediction and analysis tasks. This sets the stage for a much more efficient and accurate approach to biological research. Before we delve into the specific methodologies behind these achievements, it's crucial to understand the general landscape of what they were trying to achieve and what major findings they uncovered. Their findings give us a good perspective before getting into the nuts and bolts of how the LLMs were applied. So, let's dive into those general findings next. Now, let's get into the methods used in this research. It's important to understand that Zhou et al. Didn't develop one specific method, but rather reviewed many different studies. What they've done is shown how a whole family of techniques can be used effectively. The core idea is remarkably elegant in its simplicity, treating biological sequences, DNA, RNA, protein, as if they were text. This lets us leverage the power of pre-trained language models. Think about it, DNA, RNA, and protein sequences are all strings of letters, A, T, C, G for DNA, A, U, C, G for RNA, and 20 different amino acids for proteins. These can be interpreted like words in a sentence. To make this work, the researchers needed to develop special tools called tokenizers. These act like translators, breaking down the biological sequences into units the LLMs can understand and process. It's a crucial first step. Then comes the power of pre-training. The LLMs are fed enormous datasets of unlabeled biological sequences. This lets them learn the underlying patterns and relationships within this data, even without explicit labels. It's like teaching the model the grammar of biological sequences. After pre-training, comes fine-tuning. Here, researchers train the model on specific tasks, such as predicting a protein's 3D structure or identifying regulatory regions in DNA. This tailors the general capabilities to solve very specific problems. The authors highlight several key innovations. For example, they discuss the creation of much more efficient tokenizers, and new pre-training tasks specifically designed to address the unique challenges of biological data. Clever stuff. Of course, there were challenges. Biological data is messy. It's often incomplete, noisy, and full of gaps. Making sure these models work reliably across different species and experimental setups was also a major hurdle. Getting these models to be robust is a key focus of ongoing research. These innovative techniques and the challenges involved provide a fascinating perspective on the work. Now, let's look at the real-world implications of this impressive progress and consider what this means for various research fields. Let's move on to applications and implications. The real power of this research lies in its applications. The impact of LLMs on bioinformatics isn't just theoretical, it's already transforming several key areas. Let's start with genomics. LLMs are proving remarkably effective at predicting gene regulation, identifying disease-causing mutations, and even analyzing DNA methylation patterns. It's like having a superpowered microscope for our genetic code. Moving on to transcriptomics, we see similar breakthroughs. These models can predict RNA structure, splicing patterns, the way genes are edited, and even various post-transcriptional modifications. This helps us understand how genes are expressed and regulated. In proteomics, the implications are perhaps even more striking. LLMs can predict protein structures, predict their function, and even help us design entirely new proteins with specific properties. Break time equals 0.5s, greater than imagine creating proteins tailored to fight diseases or improve industrial processes. The pharmaceutical industry is already taking note. LLMs are improving drug-target interaction prediction, figuring out which drugs will bind to which proteins, accelerating drug molecule generation and helping to predict which drug combinations will work best together. Break time equals 0.5s, greater than this is huge for drug development. Finally, in the exciting field of single-cell analysis, LLMs are helping us analyze the complex gene expression profiles of individual cells. This leads to better cell clustering, more accurate cell type annotation, and more efficient integration of diverse types of biological data. It gives us a much more detailed picture of cellular processes. The overall effect is a massive acceleration of biological research. LLMs offer faster drug development, improved diagnostic tools, and a much deeper understanding of the biological world. We're moving closer to that futuristic lab where AI and robotics work hand in hand with scientists, driving innovation at an unprecedented pace. It's a truly transformative moment. To wrap up this section, let's summarize these key applications and then consider the overall conclusions of the research paper, and what the future may hold. Let's bring this episode to a close by summarizing the key takeaways from Liu et al.'s insightful review. We've covered a lot of ground today. The core message is clear: large language models are proving to be remarkably effective tools in bioinformatics. They're being adapted to handle biological sequences and data with impressive results, significantly improving prediction accuracy and accelerating the design of new biological molecules. The wide applicability across genomics, transcriptomics, proteomics, and drug discovery is truly remarkable. One of the key methods highlighted involves treating biological sequences like text, using specialized tokenizers to make the data understandable to LLMs. Pre-training on massive datasets allows the models to learn fundamental patterns in biological data, and fine-tuning then allows researchers to focus on specific tasks. The authors of this review demonstrated the potential of this approach across a huge range of biological applications. The implications for the speed and accuracy of biological research are undeniable. Looking ahead, the authors point to exciting avenues for future research. This includes further refinements in pre-training methods, improving how these models handle the inherent complexities and noise in biological data. Another crucial area is integrating these LLMs more closely with robotics to create fully automated labs. Imagine a future where AI-driven systems autonomously design experiments, analyze results, and even synthesize new molecules. That's the direction we're heading in. This review brings us a significant step closer to that future. Thank you for joining me on the Future Lab today. We explored how large language models are reshaping bioinformatics. Remember, this very podcast script was AI-assisted, a testament to the transformative power of these technologies. Until next time, stay curious.